So good morning everybody. I am Jay Sri, Department of Biomedical Engineering from SNS College of Technology. I am going to take the basic concept of plant growth and nutrition. Introductions. Let us see how the plants will grow. All cells of the plants develop from zygote. Zygote produces a number of cells which are organized into tissues and organs. Zygote produces a number of cells which are organized into tissues and organs. Development is the sum of two processes, growth and differentiation. During the process of development, a complex body organism is formed that produces roots, leaves, branches, fruits, flowers, seeds, and eventually they die. Growth. Growth may be defined as the irreversible permanent increase in size, volume or mass of the cell or organ or whole organism. Growth is one of the fundamental characteristics of living being. It is accompanied by metabolic process, that is anabolic and catabolic process that occur at the expense of energy, expansion of a leaf and elongation of stem, etc. Characteristics of growth. The main characteristics of growth are cellular growth, cell division, cell expansion, cellular differentiation. Growth rates. Growth rate can be defined as increased growth per unit time. The rate of growth can be expressed mathematically. Type of growth, that is arithmetic growth and geometric growth. So arithmetic growth in the sense it is periodically increase, whereas geometric growth means simultaneously increase. That is what is after mitotic cell division, only daughter cell continues to divide while other differentiate or mature. Root elongating at the constant rate. That is root elongating at the constant rate. That is what arithmetic growth. Geometric growth. Initial growth is slow, followed by rapid increase in growth, and followed by a phase where growth slows down. All cells, tissues, and organs shows this type of growth. If one plot the parameter of growth against time, it would be a typical S curve. Next, there are three types of differentiation. That is differentiation, de-differentiation, and redifferentiation. Cellular differentiation is the process by which the less specialized cell becomes the more specialized cell type. Cellular differentiation is the process by which a less specialized cell becomes a more specialized cell. Cell derived from meristems and cambium differentiate and mature to perform the specific functions which is termed as differentiation. Cells undergo. Next one, let us see the differentiation. Let us see what is differentiation. Cells undergo structural changes during differentiation. Changes take place both in the cell walls and protoplasm. Cells lose their protoplasm during the formation of tertiary elements. Plants develop a strong, elastic, and lingocellular secondary cell walls to carry water to long distance, even under extreme tension. De-differentiation. De-differentiation is an important biological phenomenon whereby cell regress from the specialized function to their simpler state reminiscent of stem cells. That is, an undividable differentiated cell becomes sometimes against the power of division. This process is called re-differentiation. An undividable differentiated cell sometimes regains the power of division. This process is called de-differentiation. De-differentiation is a common process in plants during secondary growth and in wound healing mechanism. De-differentiation is an important biological phenomenon where cells regress from the specialized function to the similar reemergence of stem cells. Redifferentiation. A de-differentiated cell can divide and produce new cells. New cells produced again lose the power of division and become the part of permanent tissue, which is called redifferentiation. That is, formation of tumor cell is the example of redifferentiation cells. A de-differentiated cell can divide and produce a new cells. New cells produce again and lose the power of division and become the part of the permanent tissue, which is called redifferentiation. Development. Development is a term that includes all changes that an organism goes through their life cycles, from germination of the seed to senescence. Okay, cell division, meristotic cell, plasmatic cell, differentiation, sensor cell, mature cell, expansion, and at last it goes to the death. Next, plasticity and heterophyly. The ability of the plant to follow the different pathways and produce the different structures to respond to environment and phase of life. In cotton and coriander plants, the leaves of the juvenile plant are different in shape from those in mature plants. The phenomenon of appearance of different forms on the leaves on the same plant is called heterophyll. So example are cotton and coriander. Next, plant growth regulators are chemical substances that influence the growth and differentiation of plant cells, tissues and organs. Plant growth regulators function as chemical messengers for intercellular communication. They work together coordinating the growth and development of cells. They could be a indole components and aldehyd derivatives and derivatives of cardinones or gases. Next, phytomodes, auxins, gibberellins, cytogens, and abscisic acid and ethylene. So let us see that phytohormones in the next class in detail. Thank you all.